Good. Okay. Well, hello everyone. I'm Joseph Yesselman. You probably heard my talks before. I'm going to be doing something a little different this time. Usually, I'll just talk about my research and how it relates to Eterna, but I thought it would be a little different this time, and I actually have a new idea that I want to hear what the Eterna community thinks of it, and possibly how this could impact a new Eterna 3D in the future. So I'm going to cover how first I came up with this idea related to my own research, the current work done on Eterna 3D for people not already familiar with it, and then how I think we could improve it. And of course, I very much would like your feedback on it. So yeah, I'm going to start pretty general. For many people, people already know this, but I mean, why do we even want to study RNA in general? And it really comes to that RNA is critical to a ton of cellular function. I'm just highlighting three really well-known examples: the ribosome, telomerase, and spliceosome, which are which are responsible for translation of proteins, chromosome capping, and removal of introns for pre-mRNAs. And these processes can only be accomplished if the RNA folds into these intricate 3D shapes to perform their function. Thus, it's very important to understand how RNA folds from primary sequence to secondary structure to then tertiary structure, so it can perform, so we can better understand how it performs these functions and why it doesn't work due to either a mutation or addition or deletion. So as I just mentioned, RNA folds in relatively two distinct states. It goes from a primary sequence to a secondary structure, and then from a secondary structure to a tertiary structure. We already have a pretty decent model for predicting secondary structure. Obviously, it's not perfect, and the work done by this lab and the Eterna community has further refined our understanding of this. But generally, it's pretty well understood. Compared to the second state, going from secondary structure to tertiary structure, we really don't have a good understanding of this at all. And part of this is because it's really critical to understand the interaction of specific parts of RNA in 3D space. So you really have to understand how things are coming together in 3D space, and that involves some really intricate modeling that we don't fully get yet. There are some advantages. We do know some general things about RNA 3D structure that can help us understand it. This is that RNA 3D structure is generally built up with these building blocks. So we have helices, which impart stability to the 3D structure, junctions, two-way and three-way junctions, which orient two helices in relation to each other, loops, which can be involved in tertiary contacts and then binding of proteins, and then, as I just mentioned, tertiary contacts, which help staple RNA in a specific 3D structure. What's really interesting about these so-called motifs are they seem to be these isolated units of folding. So to highlight this example, the simplest motif does a two base pair step, which makes up helices. If we grab the same sequence from different structures and then overlay them, you can see that even though these come from many different parts of different RNA structures, they overlay almost perfectly, meaning that it doesn't really matter where this is, it's going to give you the same shape. Not super surprising for a two base pair step, but this even holds true for more complicated things. Here's a kink turn, which is found in the ribosome, which is a very unique kinked structure. If we find another one of these and overlay it, even if it has minor sequence variation, you can see it overlays almost perfectly. And then if we find all of them, you can see that although there is more variation, the general kinkness is still there. And this really allows us to at least have guesses for what a 3D fold is going to be if it has high sequence similarity. But you can go a step further in really trying to understand more about 3D structure, and that's through design, which I'll be talking about for the rest of the talk. This first got started. Some of the first work was done by Luke Yeager, who noticed that this tertiary contact, this Tetraloop receptor, seemed to have this interesting symmetry that if you took another one and kind of flipped it upside down, you can put two helices here, and it seems like it would work. So he came up with a model, and this is before he had a 3D structure, and it worked. This complex formed with really high binding affinity, and it was later solved by NMR. And although the structure is probably not perfect, you can see it's like pretty close to what he predicted. So this kind of kills two birds with one stone. It gives us much better understanding of these rules of modularity and 3D structure, and it allows us to build new things that one day might be very, very useful. That's definitely how I'm interested, how I got interested in this. But really, I think we can go a step further. Trying to design these things by eye, as I'll be discussing later, is kind of difficult. So some of the work I've done and I've discussed with you guys in other presentations is I've come up with an automated way of designing new structures based on connecting together motifs, just like Luke Yeager did, but in like a more automated way. So the first kind of problem I was working on, trying to test this algorithm, which I call RNAMake, is this simple scaffolding problem where we have this Tetraloop receptor, just like what Luke Yeager was using, but instead of using another Tetraloop receptor, I want to connect it with a new piece of RNA and do it in a fast, automated way. And we're gonna in red on the left is the model that RNAMake would make, where the red is the new piece of RNA. And then we're going to do shape chemical mapping. And after that, the colored one is the expected shape reactivity, assuming that it was a correct prediction. And definitely take note of the very blue Tetraloop receptor, which is very protected, meaning is like, I can't because the mouse doesn't come up. Oh, you see it, but I can't see it here, so it makes it useless to me. I know, I'm sorry. I wanted to point, but I can't see it for some reason. Yeah, I don't know. You show me on the screen? Okay. Nice. So yeah, right over here. But then, okay. We're gonna double it up. So over here, right over here on the screen. Thank you, Michelle. And then alternatively, if we do not predict, if it's not predicted correctly, we expect a highly reactive shape chemical mapping in the Tetraloop receptor over here. Knowing, well, we'll know that we didn't, get it right, but we don't exactly know what went wrong. So this is a very fast screening to see if this algorithm is working properly and producing structures that like make sense. So in this first round, I produced 16 RNAs, and you can see there's a wide variety of motifs used, which is extremely interesting. That there's kind of this degeneracy in 3D space if you want to get from one point to another. Again, in red is what the RNAMake made, and then in gray is a shared structure that never changes. So we went ahead and did shape chemical mapping on all of these, and for most of them, it worked. It worked pretty well. On 13 out of 16, you can see you get something like this, where you get very good. You get very good low reactivity in the Tetraloop receptor, and and everywhere it should be reactive, it's reactive, so it looks great. But for some of them, yeah, and here's the zoom in, just so you can see. But on some of them, we get something like this, where we get a much more reactivity. It's not a crazy amount of reactivity, but more. So not sure if it actually worked. And later, RA, who just recently joined, was able to show by magnesium titration that it really was not folding nearly as well, or I'm sorry, with the gel showed that it really was not folding at all compared to something that could not fold. So, you know, we didn't fully understand why this was happening, why some of them weren't working, but we thought, you know, at the time, based on what was done in the Eterna community, maybe we can have Eterna players figure this out. And, you know, you guys are definitely really good at design issues, so this seemed like a really good problem. So this is definitely brought on by this idea of EternaBot, where the community figured out these rules for design and for secondary structure, and following a plot you've probably seen a zillion times, that we were able to encapsulate these rules into an algorithm called EternaBot, which can perform better than the existing 2D prediction, 2D design algorithms, and can do sometimes nearly as well as humans. And these were made up of a group of rules that players described, and then G, a previous graduate student who worked on this, coded them up in Python and translated them into algorithm. So we were hoping that, you know, the same thing could happen for this 3D structure. So a student named Vineet, who many of you have met, wrote up a version of this, which we called Eterna 3D, which was doing pretty much the exact same thing I described that RNAMake was doing, where your goal was to go from the Tetraloop receptor to the Tetraloop, and you were given a set of motifs, which were shown on the left, and you got to pick out which ones you wanted. And there was, even with this very primitive interface, we were given quite a few successes. Here is highlighting one by JR, which I think is extremely impressive, was able to use extremely long helices with these very bent motifs to make almost like a triangle, which I still think is one of the most impressive solutions found in general. But there were like oh yeah, and so here are the structures generated, and then we did shape chemical mapping, and we showed most of them, all of them seemed to work pretty well. So I think this was a really great success, but I think there were some glaring issues with this interface that we had. So I've just enumerated them, although I think they're pretty obvious. One, it was extremely difficult to do because there was a list of 300 motifs and you had to pick each one with no way of knowing like if it was going to work or not. So people were just like enumerating different things. Yeah, as I said, this was mostly a problem with the interface. You know, this was just a prototype, and it was extremely good, but like we didn't realize in advance that this would be so difficult. And I guess ultimately I conclude that this is a good problem for computers now, and given that I have algorithm to do this now, may not be the best use of like humans' time. So this really got me thinking. I mean, this was almost 2 years ago now, and is there a way to make a better interface for a game that would be fun for humans to play and also like scientifically useful? Part of this thinking came from some of the posts by Eli Fisker and others, who pointed out a couple things. On the left, he suggested, this is a DNA from DNA, that you can really represent. You don't have to show all the atoms. You really just need like, you know, more abstract shapes. Like it's too distracting to have everything. If you just give us the general feel of it, it would be much easier to like understand what was happening. And I think, I glanced at the figure at the right, and I thought I knew what it was, but now I'm like not super sure. But I think what he was saying was an easier way to manipulate the whole strand of RNA. Like you don't want to be putting one motif at a time. And if that's what he meant, then I am totally with him. I think this was not a great use of humans. So this, in combination with some of the really intriguing things players have made out of 2D structures, really got me thinking. Maybe instead of picking each motif, what humans would be much better at is designing a path, designing a overall shape of an RNA. Because, man, humans are really good at that, and computers are not so good at it. So I, and with other work I was doing to try to help myself make it easier to design things, I designed a very simple 3D sculpting program to make it easier for me to design things. And I started thinking maybe with some work I can make this a game. So here is kind of how it works. So here I have this Tetraloop Receptor again, and it's a very dinky interface. But what you can do is you can put points down and connect them, as you'll see. And this can represent a path that I then feed into the my program and RNAMake, and I can use it to use it as a guide to build a strand out of RNA with. So I'll show you in a second. Here I'm just showing all the things you can do with it. And again, it's like super preliminary. I mean, it's like, so you can delete points very easily. You can manipulate each point by dragging it. Pretty basic. And it's not very pretty, but it's very functional and actually made it very easy for me to draw out much more complicated things. So I think this is near the end. Yeah, and so you can manipulate it in 3D, which is nice. Okay, so once you draw something, it's then converted into, so on the left is from this program I wrote, and then on the right is a PyMol session. It's then converted into a series of points, which are then used as during this RNA building phase. And for this case, you can see it builds something that's like pretty close to what I gave it. It's not perfect because RNA, obviously, like there's only so many motifs I can use. So it can't follow the path perfectly. And that's when I really realized that there's actually something to this. Because lots of times to like find a path that's actually doable took me many attempts to like figure out what path would make sense. So here, to come up with something that we were trying to make, something that was twisted, I tried many different things because I didn't really know what twisted structure would work. And I think this is something that players would actually be quite good at. It's something computers are really terrible at. I couldn't figure out a way to do it at all. I mean, maybe someone has figured out a way to do it. And so I really thought that if you're given some general shape, and then we're asked is like, okay, for this case, I just want something that's twisted, find me a path that's twisted, this would both be fun and kind of rewarding, because the actual computation time to make one of these is only a few seconds. But whether it works or not, it might take quite a few iterations. Can
you just explain it? What was the goal here that the computer had a tough time solving?
Sorry.
Making a twisted path.
Yeah, just following the path. It sometimes can't do it because there's no motif that will get you there.
You said generating the twisted path.
You said generating the twisted path.
Oh yeah, in RNA is difficult. Yeah.
Do you mean finding a path that's actually followable?
Yes.
Oh, so
Yeah.
you can create the path.
Yeah.
And how long the computer can tell you within
A few.
seconds?
Yeah.
I can't do it. Does not
Yeah.
compute.
Yeah.
Okay.
Yep.
Oh, okay, got it.
Yeah.
So you need to have an intuition for the computer algorithm.
Exactly.
Some familiarity with it, its personality, its abilities.
You mostly want motifs are available to you. Yeah.
I see. So
Yeah.
I see. So you could
Yeah.
intuit.
Yeah.
Oh, that bend is going to be too sharp.
Yeah. Exactly. Yeah.
Okay. All right.
Yeah. Which is something, computer doesn't seem to be super good at, but a human, could get a good intuition of that
Yeah.
pretty quickly. Yeah.
Yeah. That's a good point.
Yeah. So that's an example. And then you can do some of the same really neat things that you can do in 2D, where you can draw all these really intricate things, and I think that's a lot of fun. So it not only is it entertaining, we could also make things that we could test later. I'll have ways eventually to add new tertiary context, so we could restrain the structure. Make it more compact, have more things that we could see on shape. But of course, this is not perfect. Like, since the readout is 3D structures limited chemical mapping currently, there's only so much we can actually get out, like know whether it's if it's formed properly. And of course, the size of these constructs are much bigger than the max size we're using now. So we couldn't order them on array currently. So we would have to make much fewer of them. But I still think it would be a lot of fun. So I have a quick demo I want to show you guys. But first, I want to thank Riju for giving me these opportunities. Vineet for doing the initial 3D, doing the initial Eterna 3D, and Alex, who's helping me on who helped me on all of the chemical mapping stuff. So I'm just going to show you this quick, better demo of.
There is a question from J. Anderson Lee.
Yeah, can I just? Yeah, it'll be really quick. So I wrote this is directly trying to answer Eli's point. But I have an even more simple interface. So here, each base pair is represented by a cylinder. And the green cylinder is where you can connect a new piece of RNA. And when you click on it, it changes color, so you know that's selected. And then you can build directly from there. And so my goal would be once you selected a shape, it would instantly fill in, or tell you if it's possible, fill in by calling the C++ code I wrote. And I'll just show you some of the renderings of what a larger piece of RNA would look like.
And you can do it step by step.
Yeah, you can do it step by step. Yep.
Yeah, that's actually the question from Dan.
Oh, okay.
Can players create the path and stay?
Yeah, yep, totally. That would be my point, and it's actually easier computationally to do. So, of a type, yeah. So this is what a larger piece would look like, and it's not perfect. So canonical stuff is just these red helices, and then the blue lines are non-canonical areas. So you can get a sense pretty quickly of how stable something's going to be by seeing that, and then you could keep building if you want to. So.
Even how stable? Oh, because the blue part is you want to have more red stuff.
Yeah, you have more red stuff. Yeah.
You might have an intuition.
Yeah, and then of course you can then convert it back to. I'll just show this. Lastly, you can convert back to all atom. And so you can once you're getting close to the answer, you can go back and see if this makes sense to you. This is not entirely done yet.
There's a question. How do you specify a point or direction in 3D? Like, when you're clicking on
Yeah.
something, how does it go in 3D?
Yeah. So it's in the plane that you're in. So it takes a little intuition to get good at placing it. That's why my goal would be to make it as easy as possible to have like references of like where you are in the plane. And
I feel like this problem has been solved probably with CAD software, for example.
It's
So we'll
possible.
probably just go into download SolidWorks or AutoCAD to see what the how specification of 3D points is created.
Yeah. It definitely, initially when I started, it was very difficult, but after, an hour or two of it, I got pretty good at, knowing where to put it. Now it's granted, there's probably a better way. Like you're right, someone has figured it out.
Can you now specify connection points for tertiary contacts?
I haven't done that yet, but I have an idea for to do it. Yeah.
Or a three-way junction?
Yeah. I have, I have it set up so it can be generalized to that, but I don't have it implemented yet.
What do you think would be like a either scientifically useful or just an amazing goal for intricate three-dimensional nanostructure? What would be either like an outlandish demo, like
Yeah. Well, I think the easiest ones to do would be a switch-like problems where we're doing like 3D switches instead of 2D switches.
Is that easy?
Oh, well, no, it's not easy. It's not easy, but it's easy conceptually to understand and to visualize. Where you're trying to break a tertiary contact upon binding of a ligand, or the other way around. I think those would be easy to explain how to do it, and it would be easy to come up with a solution. Whether it work or not, I don't know. So that was what I was thinking of.
Another question is
Yeah.
from Nando.
Yeah.
Any chance for AFM readouts, atomic force microscopy?
Oh,
Or cryo-EM and MR someday in the far future?
I mean, maybe. I mean, they're way too small now. But yeah, one day possibly.
There's a gap, I guess, between our ability to readily synthesize large RNAs and at the scale where you would really be able to see the design by microscopy. Yeah. You would kind of want 1,000 nucleotide RNAs.
Yeah. But of course, I was thinking, there's no reason you couldn't load the whole ribosome in here and have someone build off of it and try that instead.
Oh, yeah.
Yeah. I mean, why not? It's straightforward.
Yeah. But then again, introducing those parts.
Oh, yeah. I mean, I'm just saying, game-wise is possible.
Use the ribosome as a scaffold. And say, build a path that comes off this helix and then ends up connecting by tertiary contacts into this other helix.
For sure.
We could synthesize that 200 nucleotides piece.
Yep.
For sure. Insert it into a ribosome. And image it. I'm just trying to think. I wonder if there's a way to do that in a massively parallel way.
Yeah. Well, why not have like a place where, I mean, I don't know, that has a longish single strand area that could just hybridize.
Oh yeah.
Yeah.
Well, you want to make sure that the attachment to the ribosome is rigid.
Yeah.
And if you're going to carry out cryo.
But it would be rigid upon hybridization.
Make a ribosome where all of its stuff sticking out, all of its hairpins sticking out, have like receptors. True. Yeah, that might be the easiest. Design that has a Tetraloop.
Yeah, it's kind of like the reverse claw type thing.
So these little handholds.
Yeah.
Okay, so we're thinking about that.
Yeah.
Actually, I think Nana's question is a great one.
Yeah.
Ask another question. Do we get a better delta G estimate for these 3D structures?
At some point, maybe. I mean,
you have a scheme, right?
Yeah, I have a scheme. I mean, it matters what we're trying to make. If we're just trying to make the Tetraloop Receptor, like these mini constructs that I showed you earlier, then yes, we have a way of doing that. And that could be done, but it is much more computationally intense than just building the path. So, yeah.
Jay Anderson Lee, is it easy to make the 2D sequencing structure to 3D as a game alternative view?
No. maybe for some very small things, but not in general.
It's hard to
Yeah.
manipulate in 2D and then see the 3D update.
Oh, sorry, I didn't understand the question. Sorry, can you
It's
repeat it?
easy to map the 2D sequence of structure to 3D, meaning
Yeah.
manipulate. I think what.
Yeah.
Manipulate 2D and then like go back to 3D.
Yeah, I think that's the question.
Oh, yeah, that wouldn't be hard.
How would
Yeah.
you specify what's happening in 3D fully with 2D?
Well, I mean, if you just had like a mini map in the corner of the 2D rendering, and you just clicked on something, then and just have it reflect. I mean, the way I have it stored.
You might need an algorithm that would make a 2D version that is as faithful as possible to what you see in 3D. We don't have that algorithm now. Right now, in 2D, we have, of course, Eterna's renderer, which makes everything look like a big.
What do you mean? I have something that goes from 3D to 2D.
Oh, you do?
Yeah.
What?
Oh, you mean render the image or just make the secondary
I
structure?
suppose you make a Darth Vader mask.
In
3D. That's what your goal is.
Yeah.
You would want, and you have a bunch of branches and long helices and tertiary contacts that form the Darth Vader mask.
Yeah.
If you just render that same RNA strand and its secondary structure in 2D, it's not going to look like.
Oh yeah. No. Yeah, I have no way of doing that. Yeah.
I thought about how to do this. I think there's a polynomial type algorithm that will produce the best layout in 2D to match
like
the view in 3D.
Yeah, like a 2D production.
Yeah.
Yeah.
I think there may be a way to do that. But to my knowledge, no one has coded that. That kind of thing.
Right.
That would be fun.
Yeah.
Maybe that's one way to go.
I agree. That's cool.
And then you can export then the 2D problem to Eterna Classic, right? That's
Totally. Yeah.
Let's see. There's more questions. Nando, Janderson, Lee. The 3D structures may contain pseudoknots, which we won't be able to handle in 2D anyway. Well, I would say we should get Nando on the hangout.
Yeah, maybe.
Or Janderson.
Yeah.
You could handle pseudoknots just in the same way that Cody, Geary, and others have kissing loops. They're just these hardwired complementary sequences at the ends of loops. But I kind of want to just challenge you or other people tuning in to think about what would be the ultimate, what would be a miraculously intricate 3D structure where if you created an RNA, folded up into it, and demonstrated through crystallography or cryo-EM that you can fold up into that single shape. And then you showed it to a mechanical engineer or a biochemist, and they say, "Oh my God, I'm totally convinced that you can make any 3D shape with RNA." What's that shape? What is that or set of shapes? You know, folks have already made letters with using DNA origami.
Yeah, sure.
And what is there that's left to do that would be just astonishing?
Of just shapes or of like, I mean,
complex 3D shapes that are ideally atomically precise. And folks have done polyhedra already.
Yeah. I mean, I think of all the basic shapes, those have definitely been covered.
The basic shapes have been covered.
Yeah.
What is just kind of out of the bounds of current design methods, but is something that you think would be that we think would be so highly non-trivial. Say that the whole thing is solved.
Yeah. I mean, I can't think of any anything that's just a complex shape. I don't think has really been created yet. I mean,
would it be
like a microphone, or I mean, like anything that's like not just has very simple symmetry has not been made yet. I think the closest in RNA, the closest.
Well, but you said a microphone.
Oh yeah, I'm just saying any shape. I think it would really be like what you wanted to accomplish, right? If you just wanted to make something some shape that hasn't been seen before, that's pretty easy. That's pretty easy to do in the sense that not many complicated shapes have been made yet. I'm just saying, wouldn't you want something that was like, functional, something that would have a point?
Okay.
Yeah.
Jansen use RNA instead of protein to encapsulate drugs. Well, I think Sean Douglas.
He's already. That's probably not
as a demonstration.
Yeah, it's probably not a good use of the.
Caging drugs.
Yeah.
And I was saying things like tetrahedrons have been synthesized, and the answer is yes.
Yeah.
Tetrahedrons and other polyhedra. Actually, maybe not an icosahedron, but I don't.
Proteins are pretty good at that, though.
Proteins are pretty good. The thing that I always like about RNA in particular is that it has this you have this way to encode branched, have it look like a branched polymer, and a lot of natural shapes are branchy, like humans. We have these long arms, and then we have hands, which are branches. I'm wondering if there's something that you can't really fill out with other kinds of nanomaterials that would be trivial to set up with a piece of RNA, like some space-filling fractal-like, but asymmetric fractal. So a ribosome is like this asymmetric fractal that self into a machine. There's something that we see in nature that looks like that. Some compact space-filling thing that, at its core, is a highly branched polymer. The other alternative is to make something that's topologically very complex. So using tertiary contacts and junctions, could you make something that's like, something that's mathematically the hardest knot that's possible to make with a certain length of rope or with a certain branch polymer? Anyway, so it's worth thinking about that.
Yeah.
Make like those optical illusions and cool things
from Miami Valley. Yeah, folks have made Mobius strips already with DNA origami. That's cool. It's already been done. So we'll take suggestions from players.
Yeah.
Send a direct message to Joe with your ideas for what you want to make in 3 dimensions, and then we will make an interface.
Yeah, and then any comments on that you haven't said here, I would love to hear.
Jay Anderson is asking, in what ways does the structure of RNA affect its functionality?
Well, that's a very general question.
What are examples of how RNA structure can lead to interesting functions?
It can lead to binding for small molecules with junctions. Generally, stuff happens at non-paired areas. It can lead to catalysis. Pretty much kind of like proteins, there's a lot of structured area that leads to something happening at a very small site in comparison to the whole structure. So I guess
I'm gonna paste this link into the chat.
Oh, I don't have the chat open, but I can open that.
You have anything else open? Slack?
Yeah, I have Slack open. Yeah.
Okay, so here I have this. This is the kind of classic movie, I think, from Venki Ramakrishnan's lab at LMB on a ribosome in action. And this is an RNA-based machine that requires 3D structure, and it really looks like a robot.
Yeah. Where did you?
Oh, sorry, I just put it into the.
Okay.
Here,
Okay.
You bring that up.
Yeah.
Folks can see it. Yeah, so that's the mRNA on the bottom. So that's part of the RNA machine, the tape that's being read. And this is the ribosome, which will translate that to a protein. The RNA is mostly, the ribosome, which is the big pink thing, is mostly RNA. And the other objects that are going in and out are also RNAs. Those are transfer RNAs. And so here, all of these pieces fit together into a big nanostructure that then transits through multiple states. And it only works because everything has the right shape and can change its shape between a small set of alternative states to ratchet between those states.
Mm-hmm.
So this is going to go in slow-mo, and then it's going to speed up, and then it's going to look more impressive. So again, the mRNA is at the bottom. That's like this necklace.
Joe, is there a way to convey that in your little GUI? The state transition? The possible states?
Not currently. It's possible.
I think you brought it up. That the 3D switch is like where
Yeah.
it functions at. And I agree with you. I think that the 3D dynamics is something where we can really conceptualize designing it with RNA in a way that's not as easy with other substrates.
Yeah.
You know?
Yeah.
So that includes things like machines that ratchet, that's the protein coming out the other end.
Yeah.
This is the fast version. I think this is actually still, I think, 10x slower than real time, by the way. So if you were watching a ribosome in real time, it would process something like, I think, about 10 amino acids a second, 10 to 20 amino acids a second, about as fast as a movie playing. So it would really be, you wouldn't be able to see it with your visual eye. But also things like robotic arms and claws, for example. I think that it's less likely that we'll be able to make those with other kinds of substrates, but with RNA it seems to be.
Mm-hmm.
Anyway, so maybe that's something to think about for Eterna 3D. Like it should be, maybe it should just be, we just jump to Eterna 4D.
Yeah.
Skip a step.
Yeah, I mean, for simple things like the switches, you can just show two screens at once. You can show like what you think it'll look like with the ligand and what you'll think it'll look like without the ligand. I mean, that's pretty straightforward. But like, you know, like this dynamics or something that's, you know, quite a bit harder.
Okay.
Yep, that's all I got.
Thanks everyone for tuning in.
Yep. Bye-bye.